So welcome to this biology session. This is going to be about the first two sections of chapter 31 in the biology textbook. And in 2 weeks, we're going to do the second half of the chapter. So make sure to join us for that or watch the recording for the second half. And this chapter is about the nervous system. So we're going to start by talking about the main divisions of the nervous system. We've got the central nervous system and the peripheral nervous system. And we're going to talk about um, how impulses, signals go through your nerves, what causes that to happen, and then some more details about the central nervous system. So the nervous system takes in information and sends signals around the body. So we have the central nervous system, which is the brain and spinal cord, and the peripheral nervous system, which are your nerves. The brain and spinal cord are shown in yellow on this diagram on the right. That's the central nervous system. And then everything shown in blue, sorry. Okay, that's the central nervous system. And then everything shown in blue is the peripheral nervous system. And both of these are built from cells that are called neurons. So those are the name for the cells that build up the nervous system. Generally, the peripheral nervous system takes in information. The central nervous system creates a response, and it sends the appropriate body part, the message, back through the peripheral nervous system. The messages carried by the nervous system are electrical signals called impulses. Sensory neurons carry impulses from the sensory organs to the central nervous system. The information collected by neurons is amazing in its variety. Your eyes collect information on the intensity and frequency of light, and your ears the intensity and frequency of sound. Sensory organs in the mouth and nose detect chemicals in the food and air. Skin can sense pressure and temperature and more. Internally, you have sensory neurons that collect information from other internal organs. For example, you may feel hungry or may feel a stomach ache due to sensory neurons. Then we have interneurons. Interneurons are the neurons in the central nervous system that process the information and send out the appropriate commands in the form of impulses. And motor neurons, oops, so that's interneurons. Motor neurons carry impulses from the central nervous system to the muscles and glands. The parts of the neuron are the cell body, which contains the nucleus and has branches called dendrites coming off of it, and the axon, which is a long fiber coming off of the cell body that carries impulses away from it. The axon ends in branches called terminals. Motor neurons carry impulses from, oops, I'm sorry, I read the wrong part. So axons carry impulses away from the body, and they end in branches called terminals. The axon terminals of one neuron reach the dendrites of another neuron so that the impulse can be passed from one neuron to the other. And we're going to talk about how that works in a few slides. So we're talking about this signal called an impulse, how neurons communicate with one another. So what is this impulse and how is it created and passed from cell to cell is the question what we're going to figure out now. The impulse is a small electric current that moves across a cell membrane. A neuron normally has a different electric charge inside of it than the environment outside. And this charge is created by ions inside and outside of the neuron. On the next slide, we will see how this difference in charges is made and how it changes. So here we're looking at one end of a neuron, and we have two types of ions. These purple ones are potassium ions, and the green ones are sodium ions. So they're two different chemicals, but they're both positively charged. So active transport moves potassium ions into the cell and sodium ions out of the cell. And this creates a high concentration on each side. So these ions would like to spread out. The potassium would like to leave, the sodium would like to go into the cell so that they're spread out. But there are proteins that allow the potassium to move through them, but not the sodium. So these proteins allow the potassium to exit the cell and get away from the other potassium, and the sodium can't enter the cell. And this results in the outside of the cell being very positive, as it has both potassium and sodium ions hanging around, making it positive, and the inside of the cell is much less positive than the outside. We call this difference in charges a resting potential. And in real life, there are many more ions, and the difference is much greater than is illustrated here. I just wanted to show it as simply as possible. So this is called resting potential when there's a greater charge outside of the cell than inside of it. And the reason that happens is the potassium is allowed to leave the cell. And so you have potassium and sodium building up the positive charge outside. So, oh, there we go. That's what I said. Outside is more positive than inside. So the resting potential is the difference between the charges inside and outside of the cell when nothing is happening. When a nerve impulse happens, it reverses that difference in charges. A channel opens up that lets the sodium ions into the cell. This reverses the difference in charges. The cell is now more positive inside than it is outside. This new setup, with the inside having a more positive charge than the outside, is called the action potential. And this impulse races down the axon, changing the resting potential all the way throughout the neuron. So in resting potential, you had a higher positive charge outside than inside. In action potential, we've changed it. The sodium was allowed to come in, and now we have a higher charge inside than outside. Once the impulse passes, the cell resets itself so it can be ready for another impulse. It does this by closing the sodium gates, opening the potassium channels, and then using active transport to pump sodium ions out and the potassium ions in. The potassium ions, though, flow back out of the cell. As you just saw, they came in and then went right back out. And the cell is now again in a state with resting potential. So all we've done is we've gone back from the action potential back to the resting potential by closing the sodium gates and opening up the potassium gates or potassium channels. Yep, and now we're back to resting potential. So, a small stimulus may not be enough to start an impulse. Each neuron has a threshold, a minimum stimulus that is required to produce an impulse. There are not strong and weak impulses. There's either a full impulse happens or none at all. So how do we know whether a stimulus is strong or weak? What we're saying is your nerves are responding to something. How do we know if they're responding to something strong or to a weak signal that they're responding to? Well, the brain monitors the number of neurons that have impulses and the frequency that an impulse is repeated. In a single neuron, I'm sorry. The brain monitors the number of neurons that have impulses and the frequency that an impulse is repeated in a single neuron. A weak stimulus, such as touching lukewarm water, will have fewer impulses than a strong signal such as touching boiling water, which would set off many more neurons with very quickly repeating impulses. And we mentioned before that the impulse has to cross from the axon of one cell to the dendrites of the next cell by crossing the gap between them, which is called the synapse. The synapse is the space between these cells. The axon terminal contains tiny sacs of chemicals called neurotransmitters. These sacs reach the end of the axon terminal and release the neurotransmitters into the synapse. They cross to the next cell where they bind to receptors. And these neurotransmitters binding to the receptors starts the impulse in the next cell. So that is the end of the first section. So we have a couple review questions before we move on to section 2. Okay, so I'm going to bring up a poll on the screen, and it's this question. How does a positive charge build up the outside of the neuron in between impulses? So if you're watching the recording, just think about it, but if you're here live, you should answer this poll. No one can see your results, so don't be afraid to get it wrong. I can only see the percentage from the whole class. So let's read our question carefully and then answer it. So how does a positive charge build up on the outside of the neuron in between impulses? So in between impulses, positive charge outside, we're figuring out how does it build up the resting potential. So our options are both the sodium and potassium gates are open, allowing ions to flow freely. The open potassium channels allow potassium to leave too and go out with the sodium, or the open sodium channels allow the sodium to enter. Okay, so I see only a third of our students have voted, so I'm going to give you another 15 seconds or so. Try to get a vote in. Okay, well, thank you those of you that participated. Oh, okay, we got everyone in the last couple seconds there sent in their answers. So thank you, everybody. I'm so glad that you participated in the polls here. And it looks like we got some mixed answers. So let's look at what our options are. So nobody picked A, that's good. So both the sodium gates and potassium channels are open, allowing ions to flow freely. So that would not allow a charge to build up. If they could just go wherever they wanted to, they'd even out, no charge would build up. How about B? Open potassium channels allow the potassium to leave too. So that is what happens. The correct answer is B. If you didn't get that, that's okay. We'll talk about why that's correct. So remember, we're building up this resting potential in between impulses. So after the impulse happens, it needs to reset itself. So we need to get the positive charge on the outside. So the only way to do that is to take these potassiums that are inside and let them go outside. And now you can see we've got, a positive charge out here. In this diagram, it's +5. And that's greater than the positive charge inside, which in this diagram is only +2. So that allows that charge to build up outside. If you said option C, that's okay. This is like a really kind of complex thing. And it's just kind of really specific details, so don't feel bad. Just make sure when you study for this, you really go over that section. The problem with C, open the sodium gates to allow the sodium to enter, that would build up a positive charge inside. Okay, if we had this sodium going in there, we'd be putting more positive charges inside the cell. So that's not what we want to happen. We want your positive charges on the outside. Okay, so again, that's kind of a really detailed idea. Don't feel bad if you didn't get it. We just went over it quick, but make sure that you do read and study that page where it talks about impulses and um, active potentials and resting potentials. And you can watch those slides of this review again a couple times if you need to. Here's another question. This one's a bit shorter. So which of the following is a part of the peripheral nervous system? So again, if you're here live, you should see this poll on your screen. Please answer it. If you're watching the recording, you don't have to answer. Just watch it and think about it. So our choices are for the peripheral nervous system. We have brain, nerves, spinal cord, and interneurons. So I'll give you about a minute to answer those. Sorry about that. It should be on your screen now. You should be able to vote. And I'd like to get one last vote in here because we've got almost everybody having voted. So we'll give you another 10 seconds to get that last vote. Okay, awesome. Okay, so it looks like 67% said brain and 33% said nerves. So I'm going to go back to that screen. It was one of the very first screens, so it might have been before some of you got here, and that's okay. Okay, so the nervous system has two basic divisions. We have the central nervous system, which is the brain and spinal cord, and that is what I circled in red here, kind of poorly, but it's sort of circled. So it's the parts in the diagram that are yellow. You see the brain and the spinal cord. So they're called central because they're in the center of the body. They're not at the edges. You got, the brain and the spinal cord comes right down the middle. Then you have the peripheral nervous system, which are the nerves that come out in blue. So everything over here, peripheral, they're on the edges, okay, and all the blue things, even down, the hands, the feet, everywhere. So those are the two distinctions. You've got the brain and spinal cord making up central nervous system, and the nerves and some cells that kind of help support the nerves are the peripheral nervous system. So we'll go back. There we go. So the brain is central nervous. The nerves is the correct answer. Spinal cord is also central nervous system. It's kind of like highway exits coming and going from the brain. And that third answer, or that last answer, interneurons, those are the neurons that are in the central nervous system. So we talked about sensory neurons, interneurons, and motor neurons. Okay, so those of you that got it right, great job. Those of you that got it wrong, I think you were just not here when we went over it because that was the first slide. So just make sure you go back and go over the beginning of this chapter. And we're going to move on to section 2. So the central nervous system is made of the brain and spinal cord, like we just said. The spinal cord is the main communication link between the brain and the rest of the body. The signals from the brain go to the spinal cord, and they go along different branches from the spinal cord that lead from the brain to the spinal cord to the rest of the body, kind of like exit ramps off of an interstate that lead to smaller local roads. It also works in the other direction with signals from sensory organs all over the body coming together in the spinal cord to be sent to the brain. So that's the main job of the spinal cord. And then we have the brain itself. So the brain processes the sensory information that it receives. It has different areas to handle different information. So cerebrum handles the voluntary activities of the body, things that you consciously do, like move your legs to walk. It is also the site of learning and judgment and long-term memory storage. The cerebellum is another part of the brain. This processes the information about your body's position and information about other sensory inputs. So the cerebrum sends the command to the individual muscles to walk, but it's the cerebellum that coordinates it all so you don't fall over when you try to walk. The hypothalamus handles internal information about the body's needs, such as thirst, fatigue, and body temperature. It also coordinates the nervous system and the glands in your body. Then we have the thalamus. That's an area that receives messages from sensory receptors all over the body and relays that information to the correct part of the cerebrum. We have the brainstem that regulates the flow of information between the brain and the body and controls the important involuntary functions, things like keeping your heart rate steady and breathing. And lastly, we have the limbic system, and that's an area that processes fear and pleasure and emotional learning, and it also encodes long-term memories that will be stored in the cerebrum. So I know that was a lot of information at once, but you want to remember at least the basic idea that there's these different structures in your brain and they're responsible for very different things in keeping your body alive. And we can also look at the divisions in the brain in the lobes. So this is specifically in the cerebrum function. So I'm going to try to write, and it's going to be messy. Okay, and not too bad. So we just talked about those 6 basic structures in the brain. One of them is the cerebrum that does all of the higher thought kind of things. And we then take the cerebrum and divide it up into different lobes that also have more specific jobs. So we have 4 that we're looking at. The frontal lobe handles judgments and planning. The parietal lobe handles reading and speech. The temporal lobe handles hearing and smell. And the occipital lobe handles vision. Now, everything that the brain does, receiving signals, thinking, and sending signals, it all relies on impulses sent from neuron to neuron using chemicals called neurotransmitters. Other chemicals taken into the body can interfere with how the neurotransmitters work, and this is what drugs do. Nearly every addictive substance, so alcohol, tobacco, heroin, cocaine, methamphetamine, many other drugs, they all produce changes in the synapses that use the neurotransmitter called dopamine. Dopamine is part of the brain's pleasure and reward system. When an activity brings a person pleasure, neurons in the brain release dopamine into the synapses so that other neurons are also stimulated, giving sensations of pleasure and well-being. So different drugs cause different changes to the process of releasing dopamine, but generally, some way or another, they increase the amount of dopamine available to the brain. As a result, the brain starts reducing the number of dopamine receptors so that it isn't flooded with too much dopamine. And this is a major factor in addiction. Because the addicted brain responds less to dopamine, addicts can feel depressed or sick when the drugs aren't there acting in their system. They need the drugs to feel normal, and they cannot get as high from the same amount of drug. Because the first time you would take it, all of the brain's receptors are functioning, so they all respond to these increased levels of dopamine. But as addiction takes hold, there are less receptors available, so the same amount of drug may produce the same amount of dopamine, but it doesn't get um, recognized by the brain. Okay, and that actually brings us to the end of section 2. It was a little shorter than section 1, and we have a couple more review questions. Okay, so the frontal lobe, parietal lobe, temporal lobe, and occipital lobe are all parts of the what? Which part of the brain are they in? We got one vote right away. Good job. Waiting for the rest of your votes. I'll give you about 30 seconds. Okay, well, those of you that voted, good job. The correct answer is cerebrum. So um, that's one of the parts of the brain, and then it's divided further into these 4 lobes that we talked about. This is just a different part of the brain. Limbic system, cerebellum is a different part of the brain, and peripheral nervous system, remember, is the nerves that are out in your body. They're not part of the brain or the spinal cord. They're part of your arms and legs and stomach and whatnot. And one more question, and this one I can't make a poll just because my answers are too long, so you just need to think about it. So I'm going to read it and give you a few seconds to think about the answer. So how does drug addiction change the brain? And you only have 2 options here. Does the brain increase the number of dopamine receptors so the user gets higher every time? Or does the brain decrease the number of dopamine receptors so the user needs more dopamine to feel normal? So I'll give you a few seconds to think about it, and then we'll go over the answer. Okay, so our correct answer is the brain decreases the number of dopamine receptors, so the user needs more dopamine to feel normal. So remember that using drugs actually changes the physical structure of the brain. So addiction is a disease where the brain itself is changed and it's going to affect how the person is both in regular times and when the drug is being taken. So it decreases the number of receptors, and that means that normally when your brain is producing normal amounts of dopamine, it's not enough, and the brain feels messed up because it doesn't have the normal amounts of dopamine. So they feel sick, they feel depressed, it's not good. Um, when an addicted person then takes the drug and the brain starts releasing more dopamine, it just makes them feel normal instead of getting to the high that they may have originally gotten because the brain is trying to cut back on how much dopamine it could respond to. Okay, um, which I showed with this little diagram how All of these dopamine receptors are supposed to be open, but when addiction happens, the brain starts shutting off dopamine receptors so that the dopamine's still being released, but most of it can't be received anywhere and cause any reaction. All right, and that brings us to the end of this section. So next week we're doing a different chapter, and then the week after we'll do the second half of this section. So if you're watching this live, make sure you come back then. If you're watching a recording, check for the recording later. And if you have any questions while you're watching the recording, here's how you can contact your instructor. And those of you that are here live, I'm just going to hang out for a few minutes and stop talking and give you a chance to ask questions. So you should have, if you're on a phone or a tablet, a question mark icon you can click on and it'll ask you to type a question. And if you are on a computer, you'll have a speech bubble icon you can click on to ask questions. So I'll stop talking, you'll get the chance to answer questions. And if we've got some more questions, I can stay here for a few more minutes to answer them.